This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is the first step in the Southern Blot procedure? Answer: the first step in the Southern Blot procedure is to enzymatically cleave the DNA sample into smaller pieces. Question: how are the smaller pieces of DNA separated during the Southern Blot procedure? Answer: the smaller pieces of DNA are separated on a gel by electrophoresis. Question: what happens to the separated pieces of DNA after they are separated on the gel? Answer: after they are separated on the gel, the DNA pieces are transferred to a membrane. Question: what is the membrane exposed to after the separated pieces of DNA are transferred to it? Answer: the membrane is exposed to a labeled DNA probe that anneals to its complementary strand. Question: what is the resulting labeled piece of DNA after the membrane is exposed to the labeled DNA probe? Answer: the resulting labeled piece of DNA is a double-stranded piece of DNA. Question: how is the resulting labeled piece of DNA visualized in the Southern Blot procedure? Answer: the resulting labeled piece of DNA is visualized when the membrane is exposed to film or digital imager. Question: what is the purpose of the Southern Blot procedure? Answer: the purpose of the Southern Blot procedure is to detect and analyze specific DNA sequences in a sample of DNA. Question: how does the Southern Blot procedure detect specific DNA sequences? Answer: the Southern Blot procedure detects specific DNA sequences by exposing the transferred DNA fragments on a membrane to a labeled probe, which binds to its complementary sequence. Question: what are some common uses of the Southern Blot procedure? Answer: some common uses of the Southern Blot procedure include identifying genetic mutations, analyzing gene expression, and detecting genetic variations. Question: how can the Southern Blot procedure be used to identify genetic mutations? Answer: the Southern Blot procedure can be used to identify genetic mutations by comparing the DNA pattern of an individual with a known genetic disorder to the normal pattern. Question: are there any disadvantages to the Southern Blot procedure? Answer: some disadvantages of the Southern Blot procedure include its sensitivity to DNA degradation, the need for large amounts of high-quality DNA, and the possibility of false positives or negatives. Question: can the Southern Blot procedure be used to detect multiple DNA sequences at once? Answer: yes, the Southern Blot procedure can be used to detect multiple DNA sequences at once by using multiple labeled probes, each specific to a different target sequence. Question: what is the technique called when multiple labeled probes are used to detect multiple DNA sequences at once? Answer: the technique of using multiple labeled probes to detect multiple DNA sequences at once is called a multiplex Southern Blot. Question: is Southern Blot the only technique for detecting specific DNA sequences? Answer: no, Southern Blot is not the only technique for detecting specific DNA sequences. Other techniques include Northern Blot, which is used to detect RNA sequences, and Western Blot, which is used to detect proteins. Question: how does the Southern Blot compare to other DNA detection techniques such as PCR? Answer: Southern Blot is a relatively low-throughput method that can detect specific sequences in large amounts of DNA, while PCR is a high-throughput method that can amplify specific sequences in small amounts of DNA. Southern Blot is typically used for detection of known sequences, while PCR is used to detect unknown sequences. Question: how long does it take to perform a Southern Blot? Answer: the time required to perform a Southern Blot can vary depending on the specifics of the experiment and the equipment used. A typical Southern Blot can take from several hours to a day to complete.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.